I will always respond eventually. So this isn't your only chance to talk to me if you want to talk to me.
That's
Maybe
true.
In person.
The only chance by a fire.
There we go. And I'm not sure. At times I feel like you know it's going to seem silly to say this, but I'm not expert in everything. But a lot of questions here that I would not be able to answer, especially when it comes to implementation stuff. So I'm looking around for some friendly Numenta faces who might be able to help me. You're it, Matt. You're the only one. So
Well, if anybody wants to jump in, and
we have a question about some implementation detail I don't remember or something.
Yeah.
But anyway, there's no agenda here. So we can talk about anything. If anyone has any questions, we can talk about business issues. We can talk about technology. Yes, we can start off.
And I guess this would be something simple. And you know, why left with
Let's
from
hope
2 days
so.
ago.
Let's hope it's simple.
About how is the time series implemented in NuPIC?
How's time series or time actual time?
Well, how is time modeled in HTM? Because at a certain point I had the feeling that I understood something from the presentation 2 days ago, and then you kind of destroyed that for me.
Oh, I destroyed it for you. Well, this is a really good question, actually. So let me try to address it at a couple levels and see if I catch what you're interested in. First of all, the model like the HTM temporal memory model is a model that's a predictive model. So it says at any given point, it's going to predict what's going to occur next. Now, there's actual no time in the system. It's just saying this is what's going to occur next, and if we don't give it anything next, the system just sits there in that state. I mean, that's the way the HTM implementation is. So, but what it does is it predicts what's going to happen next. So when something X happens, it can say that was right or wrong, and then it can predict the next thing. So it's an inference model. So if you're trying to infer, like you're trying to learn sequences and then recognize sequences, you actually don't need to encode time itself. All you need to do is make the next prediction and say the next thing that comes in, does that match it? So it's just predicting one data point at a time into the future, but it does it in a very sophisticated way. So that's for inference. I don't need to actually encode time per se in that kind of model. You just need to encode transitions, right?
Isn't that right that you feed HTM with a time series?
It's a time series, yes, a sequence.
And is that vector, that sequence, which is coming in parallel from a good number of sensors, right?
Yeah.
Advancing through.
Yeah, through time. Or I didn't.
I mean.
We may every record.
Columns.
Imagine you got a whole series of records, right? And each record is sequential in time, and it's just learning that sequence, right? I'm trying to make a distinction, which you may not be making, that we don't actually encode time itself. We don't say, oh, this one occurred 30 milliseconds after that one, or this one occurred. You know, there's no sense of like absolute time.
No, not the absolute.
Okay, so a series of records are fed into the models, and it just learns those transitions. It learns that sequence, and then when you feed it another series, it basically matches to all the previous things it's learned at once, and it's constantly trying to predict what's going to occur next. So from an inference point of view, that's all you need to do, is you just need to learn the sequence. Each time I say learn the sequence, it doesn't learn just a sequence. It's doing this in a distributed fashion, and all these sequences are sort of in the same memory structure. So it's constantly predicting multiple things at the same time. But that's, you just basically feed it in a sequence, and then when you feed in a record at a time, it just says, can I infer what sequence this is? And basically, can I make the correct prediction of what's going to occur next? And keep track of the high order stuff. There's a separate problem. I see a hand over there. There's a separate problem which comes back in playing back sequences, recalling sequences on their own, like the motor behavior. And we haven't really dealt with that yet, right? So none of the stuff we have right now actually plays back sequences. You could do it artificially, but that would require some sort of the system on its own has to generate the next event, and the next event, and the next event. But all if you're doing is inference, prediction, and anomaly detection, you wait for the data to come in, and one at a time. Does that help you? No.
To some extent, yes, but there were some pictures at that time when there were a number of so it was on the connections on distant connections.
The distal connections, yeah.
Also the one closer to the body of the
proximal connections,
proximal. There are a number of dots in there.
You're talking about some very old presentations, or?
Sorry?
You're talking about some old presentations?
It was one of the presentations which was
recent?
Friday.
Oh,
yeah.
Friday. Oh, okay.
And so there are a number of dots, and I'm curious if those dots were part of the time search.
Okay, yeah. So we there's a proximal synapse, proximal dendrite on a synapse, and this gets the feed forward pattern, right? And that's not encoding time. And then or non-coding sequences. It's these distal dendrites here that we call them coincidence detectors, right? And these are getting like connections from the other cells nearby. So basically, these are saying this cell is learning to predict when it's going to be active based on the other cell's activity nearby. So it says, if I recognize the state a moment before I become active, and then I become predicted, and then I'm primed to fire. These are encoding the sequences. This is encoding the transitions. These cells.
Okay.
This is all about just learning predicting when the cell is going to become active based on some very high order, or maybe not some very very sparse high order state that says I'm at the third, the 23rd note in this melody. I recognize that I'm part of the next note type of thing.
So those dots,
these synapses, yes,
are related to, or they become active or not, depending on what the step 10.
Step before.
Yeah.
Or some prior state right before.
Okay.
Right. Yeah. So these basically, you remember, you got a huge number of cells nearby, and at any point in time, due to feed forward input, some of the very sparse number of these cells become active, and as soon as they become active, they say, who was active nearby recently, and I'm going to try to make connections to them, and that becomes my prediction. It basically cell says, if I can see those things nearby, that they were just active before I became active, then I'm going to predict I'm going to become active. So this is the transition memory, if you will, the temporal memory. This is not.
And this question came in my mind kind of related to what I think in health sciences is they call an audio memory.
An audio memory?
Something which is, I don't know, a few seconds, they say. So we have in our memory the melody in the past few seconds. Or maybe even more.
This is a separate topic. If we start talking about in biological brains about time scales at which we can have hold things in our head or keep things active, or how many sort of a temporary memory, it's a kind of a separate problem, a separate task here. We're not really dealing with that. We can talk about that, but I think the basic thing, your basic question about where is time stored in this stuff, it's stored in the majority of the synapses that are on those active dendrites that are the distal synapses, and those represent basically transition states in the sequences. And there's a whole series of other things I was trying to get to with my answer earlier about real time, like how would I play back something with proper cadence, like a melody. How does the brain encode that? Those are separate questions. But right at the moment, the simplest thing is to say, look, where do we learn the transitions in time? Where's the sequence stored? It's stored here. Each cell is learning on its own when it becomes active. What are the things that predict it? And it can do that in hundreds of contexts. And that cell, any cell, of course, is reused over and over again in many different representations in many different temporal contexts. But it's always the combination which is unique. That was part of Subutai's talk. That you only got so many cells, and if you only, and some percentage of them active over, they're going to be used over and over and over again in many different contexts. But it's the combination which is unique. So that's why the whole memory is distributed. This guy doesn't know what anyone else is doing, but he just says, when I become active, what are the contexts I become active? So if this was participating in a melody, it could be participating in many different melodies, many different points in different melodies, and these recognize those different states and all those different melodies in some sense. I saw your question. I think next I'll get to you.
Could you give us your 50% understanding of layer 5.
We recorded a talk about this a while back, the 50% layer 4. What was the name of that talk? Sensory motor.
Yeah.
Yeah.
At a meetup.
Yeah, we did that, and it's not a short talk.
Yeah, I remember you saying that, the primitive subcortical areas already know how to do this stuff, and it grows. It just grows associative connections.
Well, the basic it's subcortical. A region of cortex is sitting on top of a whole bunch of other stuff, and it doesn't really know anything about it. You have to imagine neural tissue is dumb, right? There's no smarts anywhere. So this neural tissue is sitting there, and it doesn't really know where it is in the hierarchy. It doesn't know there is a hierarchy. It doesn't know anything. It's just a bunch of neural tissues in there. But what it does is it learns how to affect behavior by connecting to something else below it that's doing behavior. That could be, if I was looking at a cortical hierarchy, here's two regions of cortical hierarchy, and then here's some old brain stuff down here. A layer 5 cell here can project both down to the layer region below it, and it can also project down to the old brain. It basically says I can try to control anything below me, right? And this guy's doing behavior, and he's trying to think below him, so it's done in a hierarchy. But the key to the whole motor thing in a hierarchy, at least it felt that way to me, was understanding that this region of cortex is modeling everything underneath it, not knowing what it is. And it tries to build a model of how all this stuff behaves, and then it projects its layer 5 cells to the cells that are generating behavior down there and auto-associative links to it. So it's basically saying, I don't know what you're doing down here. I don't even know what I'm projecting to, but I can auto-associatively link to control someone else's behavior. So this region can control this guy, and it can control this thing auto-associatively. So I can walk through it more, but I have to get out that talk. But that was the big insight for me, was that you can do this auto-associative linking, or not auto-associative, associative linking. And this guy models the behavior of this thing, and then it learns to control it, and then it can control it in new, novel ways. It basically says, I'm learning what this lower behavior is, and now I'm learning to invoke it when it wouldn't have been invoked otherwise. So I can't go through that whole talk here, but
I guess maybe my question is, what about the 50% that?
Well, what I don't understand, what's part of it I'm confused about?
Yeah.
The part I'm struggling with right now is, I'm not right now. The part I'm struggling with when I think about this is what we know is that we know that here's cortex. We'll just call that cortex. If there was a black pen, I'd really like that. There is no black pen. Here's cortex.
Somebody get Jeff a black pen.
It's okay. It's okay. I have this thing about colored pens. I like it, but only occasionally. We know, I have a pretty good idea how this guy can learn to control this behavior down here, whether it's other cortical regions or other things. So I have a pretty good sense for how this guy can learn to control that. And I have a pretty good sense how it can learn to generate sequence of these things. What I don't know yet, and I'm struggling with, is how does it decide what to do? How do you introduce the goal-oriented behavior to that? And what we know, what neuroscience says about this, is that the cortex presents, there's another part of the subcortical thing called the basal ganglia, which is, literally it means lower stuff. Basically, that's what it means. Ganglia are just like groups of neurons. So there's a whole bunch of little things that they have go by a whole bunch of different names, but they just group them together because they're kind of small and they're all together. And what happens is, what the general belief is that the cortex presents to the basal ganglia a set of possible
alternative choices.
Behaviors. Yes, like these are things you could do, a union of them. All right, and the basal ganglia chooses one through some magical mechanism, and tells back to the cortex, execute that one. So there's a lot of existing psychology and other theory about, how we evaluate behaviors and how we choose behaviors and how we learn behaviors. And anyway, it's just, and this pathway is really complicated. It runs through all these little regions, and then it goes back to the thalamus, and it's really messy. And although there's this basic idea that the basal ganglia decides which to execute, there's really almost no real detail you can stick your fingers into or your teeth into and say, oh, that explains it. It's just like kind of mysterious. And so what I struggle with is, how is it that we who are trying to model cortex, how is it that we want to, what is the desirable outcome? How are we going to, what kind of problems can we set up where we can structure something where I have a problem where I can pick the right answer? It's just, I can't even articulate it very well. But we sort of like have a lot of the mechanisms which are in place, at least in my head they're in place. They're not encoded, but they're, but then this idea of how do you generate, what is the function you're trying to optimize? How are we going to do that? What kind of subcortical structure do we have to design to do this? Because apparently the cortex itself doesn't do that. And it's that kind of stuff. There's also another problem which I'm struggling with. If I look at the regions in the layers in the region of cortex, so you got 2, 3, you got 4, you got 5, and you got 6 down here. And these cells that generate the behavior in layer 5, those are the ones that project subcortically. But they're complex. They also project up the hierarchy as well. They receive input from layer 3. They also receive feed-forward input from layer 6 to layer 5. So there's a lot of circuitry here that I don't understand. And I wish I did understand. And you read the literature, I can't even get some basic things. Like there's a very common, you'll say layer 3 projects to layer 5. You can read that in any neuroscience textbook. Well, you say, okay, well, where do they project to the distal synapses or the proximal synapses? I can't find that. I don't really understand the relationship in layer 3 and layer 5 exactly. It looks to me like layer 5 and 6 are almost repeating what layer 4 and layer 3 do. Is that the feed-forward input that comes into a region goes into layer 6 and projects to layer 5, and the cell responses here mimic the ones we see in 4 and 3. It looks like there's almost a parallel path here. This is almost duplicating this, and so it's somehow, which is, there's a lot of questions here. It just hasn't gelled yet. So you asked me to explain things I don't understand, so it's going to sound nice.
It's the high value stuff.
Well, it's enough. There's enough there that it's not like, oh, this is impossible. It's always like, oh no, every once in a while it feels like you're really close to getting it, and you're like feeling really close to understanding it, but then it slips away from you.
On the basal ganglia,
yeah.
A naive implementation would be just an objective function, right? Some generic reward or punishment.
Yeah.
Do you see that working, fitting in?
Well, obviously, we'd like to be able to do that, right? We'd just like to do something really simple. like today, we don't really have emotional states in our models at all. We don't plan on having. We do something really simple. We can turn on learning or speed up learning, slow it down, things like that. Sure. I mean, yeah, we don't have to model basic ganglia. I have no intention to model basic ganglia. It does all these weird stuff, and it's got all kinds of stuff in it. It's like modeling 20 little things. But still, if I could just, sometimes I feel like the thing you need to solve a problem is a very, very clear problem statement. And often the trick to really solving hard things is to get the right exact problem statement you're trying to solve. And it's actually hard to do that. And so I'm struggling with that here too. I'm trying to struggle like, what would be the minimal problem we could solve that actually gets to the heart of the problem? And hopefully it would be something very simple. But it's complicated. It's not. There's a lot of pieces that are moving around here. I can't really talk about it too intelligently. A lot of the pieces are in place, and it feels like, oh, it can't be that hard, and it really can't be. Nothing in the cortex is really that hard. These are all stupid neurons, and they're all just doing simple things. Anyway, so I've been playing around with different models of different connectivity between these layers and understanding 5 and 6 versus 4 and 2, 3, and trying to figure out what's going on exactly here. And I got pieces of it. I got a lot of pieces of it. I got about half of it. I think it feels like I got about half of it. That's why I wanted to say that. Sure, Fergal, that's
yeah. I think I actually have an idea about how to do it. So
wow. Well, all right. I like that.
So that's what my demo was going to be about, but I think it'd make more sense if I if you like. If
I don't know.
run through it.
This could be a very long. Is it something
No,
short?
it's short. Yeah.
Really?
Yeah.
Okay. You want to give it a shot?
Yeah.
Go for it.
No.
I have trouble believing it's really that
Yeah.
short, but
We'll go short.
I mean, it's like saying, "Oh, explain how layer 3 works." Oh, it's really easy. This is
No,
story, you know?
yeah, no, it does all of that. Yeah, I ran this past two battalion UA and a couple of other people. So.
Okay.
Okay.
So what is it exactly you're about to
Okay.
explain?
Well, first thing is just.
Or you
like-
want to transfer my?
I'm just going to give him this one. Your star.
Cheers. Okay. First thing is just the basics about how all the rest of it works. So it's just essentially, this is completely neuroscience, right?
Okay.
But can be directly translated into algorithms.
Okay.
Okay. So I'm just going to be talking about cells for the whole thing.
But okay,
Right.
but you got to in my mind you have to put it in the context of sort of a theoretical explanation.
No,
first,
but
right?
yeah, but it is. That's
Okay.
exactly what it is. Okay, so the cells basically start off at a resting potential, which is minus.
Which
6.
cells are we talking about?
All neurons. Sorry.
To hold them back up.
All
Sorry.
neurons.
Yeah.
We're not just
But-
talking about layer 5
No,
cells.
just pyramidal cells I'm talking about.
In all
Okay,
cases.
yeah. Okay. So.
This is a generic
and
cell.
It's just generic how the activation works. Okay. And
But you're going to talk about how it generates behavior.
yes. No.
Okay.
But you need to just follow it.
Okay,
Okay.
okay.
Right. So and each cell gets a certain signal which comes in on its dendrites.
Yes.
And
Which
creates a-
dendrites? We're talking about
all the dendrites. Okay. So there are
well,
two different mechanisms.
Yes.
Okay. But they both generate current that goes into the cell body eventually.
Well, except the distal ones generate a very step function,
Correct,
and the proximal
yeah.
ones are more of a linear
Yes.
function.
But between the two of them, they're injecting a current which rises, which causes the voltage to rise, and then the cell fires.
Okay.
Okay, right. Now, if you add up the current for different cells, because they each have different sets of inputs,
Yes.
then they all have different slopes, so they all rise at different rates.
Yeah.
Okay, now, if these cells are nearby each other, then the first one to fire will cause inhibition of the other ones.
Yes.
Okay, so now the activation of the cells, the current that comes in comes from both the proximal dendrites and the distal dendrites,
Mm-hmm.
and they're added together over a
Yes.
period of time, leading to this current. Okay,
Yeah.
Right. So over a time step, you can simplify that by just saying that the distal inputs add up to a certain starting potential. Okay.
It's just a jump up.
Right. Yeah. So they basically have a head
It
start on the other ones.
depolarizes the cell.
Okay. Right. And if the inputs are the same, then two cells like that, this one will fire first and inhibit the other.
That's basic to the.
That's just the basic thing
basically
about how all these
that's
things
already,
work.
That's
Okay,
already in
that's-
the model here. Right?
Okay. Right.
That's
Okay.
how we end up with sparse representation, because the predictive cells fire first and then you look at
Correct.
the other guys.
Correct. Okay. So the model that I have for how a column works is that there are two types of cells. Okay. There's the normal pyramidal cells. Okay. And there's this cell here, which is actually a cell complex that's wrapped around the column of inhibitory cells.
Yes.
Okay. Right. Now this has a cell down here, which has a
That's
proximal dendrite.
that's what we would call.
That's like the SP.
It's like the
cell.
SP
Right,
special
okay.
code. By the way, we now know that there are inhibitory cells that do exactly this,
Yeah.
and they're tuned. So
Yes.
that's part of the SP.
Okay.
Okay. So
Now-
far we're on the
okay,
same track.
right. Now, in addition, each of these guys has its own dendrite, and they all respond to the same feed forward. Okay. Yeah. So.
Up to a point, but anyway. yes.
Okay. Yeah. No. Absolutely. So what happens is that, assuming there's no predictive input at all,
Mhm.
okay, all these cells are all at 0, are all at minus 60.
Mhm.
Okay. And they all get the same feed forward input, and they all have approximately the same response to that input. Okay, it varies slightly. But we know that this cell here, the inhibitory cell, has a faster response to the same
Yes.
input as any of these guys.
Yeah.
Okay. So that means that it's going to have a higher slope than any of the individual cells inside.
Yes.
Okay. So it will fire first.
Yes.
Okay, right. Now when it fires, it sends a signal outwards to its brothers and sisters, the other inhibitory cells, and they all get killed off. Okay.
Right.
And somewhere over here, before that signal reaches it, the same thing happens in another column. Okay.
Yes.
And that sends the same type of signal back this way,
Uh-huh.
and that means there's no activation in here.
Yes.
Okay. So these cells are now protected from being hit by an inhibition
Yes.
wave
Yes.
from elsewhere.
Yes.
Right. Okay. So
far, this is exactly
yeah.
HTML,
Okay.
right?
Right. Exactly.
Right.
Now, these guys are continuing to get the same feed-forward input.
Yeah.
Okay. Which means they all get all the way up to the top here. Okay. And they all fire one after another.
Who fires one after another?
All of these cells.
In sequence?
No, they just continue because they're never going to get stopped.
Yeah, no. You said
There's-
one after another, so I'm sorry.
Yeah, sorry. The fastest one will fire first, then the second fastest, etc.
Okay.
Okay, right? And that is what causes bursting,
Yes.
right? Okay, right?
Right.
Okay.
So far,
Right.
same
Okay,
from
so
the
that's with
beginning.
no prediction, you always get
Yes,
bursting because
yes.
this happens. Okay, right.
This is
Okay.
how the SP works, exactly.
Correctly. Right.
Okay.
Okay, and the first one to burst may cause some signal that makes it a learning cell in the current thing.
Well,
That's
what
all.
we basically happens is that all these cells still try to inhibit one another. So they burst, but then very quickly, one of them wins out because there's still those inhibitory cells that are going to inhibit one of them. So one of them's going to win. So we basically burst followed by a single cell being active.
Okay, well, I don't model that at all. I say they all fire.
They do.
Okay, right, and that the one that fires gets the longest to learn or something like that.
Okay, it's the same thing, right?
Something
But
like
basically,
that. Yeah, but it's a differential thing. But these guys all fire effectively.
Okay.
You know, for the whole rest of the time step. Okay.
But they're not learning. So what's the point?
No, but they are because they're learning to feed forward, right?
Okay.
Okay. So
You're going to bring this back into behavior, right?
yes, absolutely right. Okay. Now,
Because all we're doing so far
Okay,
is
so-
doing the spatial pooler.
yes, but this includes the temporal pillar as well. This is all one model
Okay,
that
we haven't
combines
gotten
the
there
two.
yet.
Okay.
Right?
Right. So this is with no prediction.
Yes.
Okay. Now, if you do have prediction, then one of these guys is here.
Yes.
Okay.
It fires first. It beats the
Right?
inhibition.
Correct.
It beats the
Yes.
inhibition, therefore it fires before the inhibitory cell fires. That's why one cell wins and no one else in the column
Correct.
does.
Right. And
Yes.
there must be some mechanism in here that when this guy fires, he kills off all of these
It
guys.
is. It's very
Okay.
well known.
Right. Yeah.
That's what I was saying
Okay.
earlier. Even after the cell
Yeah.
fire bursts, one of these guys will win and inhibit the other guy. So it's just, that's a general
Okay.
principle.
Yeah. Well, my model basically says that if none of them fire in here, then this is switched off. Right. Okay. And they all get to fire. Okay,
That's
and
right.
what
And
causes burst.
initially, that's right. That's the bursting.
Okay, so you think that happens at a later point in the time step, basically, that they get shut down.
They burst, and
Yeah.
then
And then they shut down.
then one wins, and then you start training on that one.
Okay, right. Well, I don't understand how you can.
It's
So
a-
if they fired, then they fired, and that's it.
No, because there's still this mechanism that says when a cell fires, it inhibits all its neighbors, right? And yet what we're trying to do is what happens is the column activation occurs first before that happens.
Yeah.
Then all of them fire, but then once one cell fires, it inhibits its neighbors. So this whole bursting idea is not something that's stable. It's a temporary thing.
Yeah. Okay. Well, maybe in a repeated step or something like the same thing happens
I don't
again,
know.
then they don't fire again or something like that.
Okay, I
But
don't know.
If they all
let's get-
fire, then
let's
they've already
bring this
fired.
back to behavior.
Okay,
that
so
was the question.
okay, so with the predictive thing is really important, right? So I'm
Okay.
going to add in prediction now. So this cell, when you get a prediction,
Yeah.
okay, that's good enough to beat the inhibitory cell, which
Yeah.
is usually better than any.
Yes.
Any
Inhibition is
pyramidal
faster
cell.
than excitatory
Yes,
cells,
yes,
but
yes.
if the excitatory cell has a head start, it can beat the inhibition.
If it's really good. Right?
Well, it's
okay,
already
if,
got
it's,
head
if
start.
it's sufficient. If
Yeah.
it's sufficient, right?
Yeah.
Okay, so something down here, for example, is not going to beat it.
No, but
Okay.
the genetic action potentials I think it was like 30 milliseconds, 30 millivolts, something like that.
Yeah.
It's a big jump.
Yeah, okay. Right, so if you have a predictive cell, then you get a highly sparse
Yes.
representation. Okay?
Yes.
Right. But you get the same columnar level activation.
Absolutely.
Okay, right. So that's simultaneously SP and TP, but the
Well, we haven't
important
talked about temporal pooling yet.
No, the old TP, the TF.
Oh, don't
Right,
use that.
okay, yeah,
That's
okay,
the temporal
right.
member. That's
okay,
the,
yeah,
yeah,
okay,
right.
right. But this is the basic CLA, all done in one go. But the
Yes.
important thing is the first thing that happens is the predictive recognition, right? Okay.
Okay.
If it's good enough, if there is one
Yes,
cell,
that's still
that happens
that's
at the
the
beginning.
basic temporal memory algorithm.
Yeah, but this happens first. That's the important difference.
That,
okay.
you know, that's the way it's always been. If this cell's been depolarized because it's been predicted, it fires first, and it shuts down everyone else. That's the way the temporal memory works.
Correct, but in the implementation, that's missing, right? So
I don't think it is.
okay, right.
That's how it works.
The other implication of this is that if you have bursting cells, if you have the column bursting, then all of these cells here learn the feed forward that matched the inhibitory cell.
Yeah.
Okay. So they tend to converge in their own dendritic signatures to whatever the inhibitory cell, the column cell, represents. Okay. But if you have predictive context, sufficient predictive context, then that cell will specialize on this feed forward input within that context better than the other cells will. Okay, so they'll learn to specialize on the feed forward.
Okay.
So you have predictive assisted spatial recognition.
How does this play back into behavior?
Okay. The reason it does is because the temporal pooling happens when sparseness is maintained. Okay, and the cells learn to tell the difference between sparseness and bursting in L4.
Yes, obviously. That temporal pooling basically relies on prediction. You only want temporal pool when it's predicted. That only occurs when it's sparse. You don't want to do temporal pooling when it's bursting.
Yes.
Okay, so far we are totally on sync. This is the basic HCM theory.
Yeah, so in L4,
All right.
you have columns here. Okay.
Yeah.
Right, with multiple cells in them. Okay, and in the case where those, let's say it's a NuPIC sized region of 2,000 columns, 32 cells per column. Okay, in that case, when you have correctly predicted transitions everywhere, you have 40 cells active.
Yes.
64,000.
Yes.
Okay, right. And when they're bursting, you have 1,260.
Yes.
Okay.
Yes.
Now, up in L3, you have these cells which have learned to.
Fergal, I'm gonna challenge you. I don't think you're heading towards explaining how layer 5 works. So far we're talking about temporal pooling and how temporal pooling is occurring in layer 3.
Yeah.
And we can talk about that, but so far you and I, this is exactly what we've laid out before. So I don't see any difference here, but we can go into talk about temporal pooling exactly how it works, because we're in the process of doing that right now. But I don't see you're going to bring this back to layer 5 and the behavior, the question that I originally got, which you said you had a short answer to. So I'm being honest about it. Look, come on.
Okay, no, but the thing is
I don't want to turn this into
when I explain this,
talk.
the same thing happens. As you said, it's a mirror image of
Yes,
it.
yes, I got all that. And that's why I said the whole thing is repeating itself down below. The question is, how do I understand?
But what is it repeating is what I'm explaining. Right?
Okay, let's try to tell me in 20 seconds.
Okay, so in here you have a really poor, low volume signal that's going up into L3. Okay, there are only 40 activations going up into L3.
Very specific.
Very specific ones, right?
Yes.
Now, out of that, only a very small number will be connected to those 40 cells, any significant number of them.
I mean, in layer 3?
In layer 3. Right?
We can debate that, because I think that's not true anymore.
Okay, but no, in this model, you have a very small
that's
number
where
of cells.
That's where we started with, but I don't think that's true anymore.
Right, okay. And
all right, really, tie it into layer 5. Come on.
Okay. Now what happens is that these cells are connected to the very small number of these things. And when you get transitions in here, the signal is so poor that those cells are basically, they're going to be connected to several of these things that don't change, for example, okay? And they're going to also inhibit their neighbors so that their neighbors aren't learning any of this current set of transitions, okay? So they're going to be an advantage to
How's this relate to
recognizing.
How does this relate to layer 5?
Okay. What happens is that when you have sparseness, you get temporal pooling in layer 3. Okay. When you don't have sparseness, that's disrupted. Okay.
Exactly.
Right. When it's disrupted, you get non-sparseness in layer 3 as well. Okay?
Yes.
And that gets transmitted down to layer 5. Okay. So basically, what happens is that the system is trying to have sparseness everywhere. Okay.
That's the predictive models. Everything is trying to do.
It's trying to have sparseness everywhere.
Yes.
And when it can't do that, it causes bursting, and that propagates everywhere. Okay?
Well, that's not necessarily true. And we can debate about that, because it's not clear. The way we're propagating between layer 4 and layer 3 is through the inhibitory cells, and it's not clear that those actually go down the other direction, but it might.
No, but what happens is that when these
All right.
cells, if there's bursting down here, that causes bursting up here, which basically
Yes, but what's
kills
going on
these
in
off.
layer 5 is not clear
Okay.
in this regard.
No, but layer 5 is doing the same thing, but it's getting predictive input from layer 3. Okay, and what it's trying to do is generate behavior that maintains the sparseness in layer 3. Okay, right? So it's generating behavior, and it learns to associate whatever it's getting from layer 3 with it's
what
either
is-
sparse or not.
But what are the driving functions for layer 5 cells? You can't understand this if you don't understand what's going on in the proximal synapses in layer 5 cells.
Okay, layer 5 is getting signals from.
It's not clear exactly what's getting signals from. Further, I invited you up here. I think you can do it. If you want to do a longer talk on this, I'd be happy to do that. But I don't think that's the purpose of this thing right now.
Sure. Okay. Well, I think that the key thing is the propagation of sparseness. Okay, and that layer.
Well, I
5
guess in
will-
general I would agree with that. In general, the
Yeah.
whole idea here is that when you have sparse representations, it means you predicted the input correctly.
Yeah.
That is what you want to use to propagate to do pooling on the next level.
Yeah.
If you can't do that, you pass up the noise to the next level. I wrote that about that in
Yeah.
On Intelligence
Yeah.
as part of this whole theory here.
Yeah.
The question
If the
is
behavior
exactly
is-
how
if
the question is exactly how that works in layer 5 is not clear. It's not clear that it's not clear that the same sort of propagation occurs in the columnar sense down to layer 5, or if it's doing its own thing, and it's not clear what the layer 5 proximal inputs are. So what is layer 5 modeling that layer 3 can be the sort of predictive signal for it? So look, I think this is a much longer conversation. I don't think we
Sure.
should turn this little session into that. And I'm not against talking about this in
Sure.
depth, but I
Yeah,
think
well,
that's
I think that layer 5 is getting a copy of the incoming sensory motor thing from layer 6. Okay, it's also getting a representation of whether or not there's a stable representation in layer 3.
Yes.
And what I'm saying is that it's generating behavior that it associates with keeping that cycle from becoming non-sparse. Okay.
it's trying to generate behavior that leads to prediction.
Yes.
I agree.
Which is
totally
which
agree.
it interprets as either a sparse signal coming from
I
layer 3 or not.
agree 100%.
Yeah.
That's
Okay.
what I wrote about this in On Intelligence. The goal of behavior is to try to make predictions come true,
Yeah.
or make things more predictable.
Yeah.
But the question, I still use the question about how do you evaluate multiple behaviors and what kind of system design to do all that. I still back to the same
Okay.
problems I had
Well,
before.
My suggestion is that the answer is that in the same way that this is sparse, if this is sparse, okay,
Yeah.
then in layer 5 it's getting a similar sort of either sparse or non-sparse signal from layer 6, okay, which is
Yeah.
coming from the sensory motor side,
Yeah.
okay, and it's trying to also maintain this sparse stable representation coming from layer 3. And so its representation, when everything is working, is a sequence of very sparse representations in layer 5 that are generating very specific behaviors
Yes, I
that
agree.
are in a very small subset of possible behaviors that will maintain this
I
feedback loop through the world.
agree. Although the
Okay.
details there are the hard part, and that's where I'm struggling. I mean, the basic idea is I agree with you. It's
Yeah.
that's.
But I think that this
I have
thing
a
where
question.
the weak
Silver
signal
tie.
is the thing that does it.
Do layer 5 cells get input from apical dendrites?
From layer 1 as well.
From
yeah.
layer
Well,
1?
they get input to layer 1, and that I think we can explain. That I understand pretty well. But
Excuse me, that has to be a part of this behavior.
Yes,
It's not
yes.
just
Yes.
this layer.
And that's
three,
also
layer
important
five.
for layers.
It has
So-
to be feedback
Yes.
generating some.
Yeah. So what I think's going, I think I've talked about this before. What I think's going on there is that you have, and this is part of the stuff that I understand better than the part of the stuff I don't understand. If you imagine here's two regions, and in general, and this is hierarchical, in general, this guy will be more stable than this guy. For all the reasons we know, it'll be more stable than this guy. And so as I go through sequences here in layer 5, this is through time. If I go through sequences, these are sparse distributed representation sequences in layer 5. As I go through them, all these layer 5 cells have apical dendrites that go up towards layer 1. They're all getting feedback from this region above. And essentially what will happen is as I go through these sequences, and there's a stable representation in layer 1 coming from above. So this is like a higher level concept. It's stable. And I'm going through sequences here. Every one of these cells can be associated with this stable representation. So in the future, now if I just present this stable pattern here, it will invoke a union of layer 5 cells to become depolarized. And what that union represents, in some sense, all the possible states of all the sequences associated with this thing. So you might think of it as multiple behaviors. So you would say, like, my goal is to, the example I use is like walking in a house. My goal is to go from the kitchen, get to the kitchen. I'm hungry. I want to get to the kitchen. And of all the times I've gone to the kitchen, I've basically associated that with multiple sequences here. And these are like, you can think of these are like the different steps I had to do to get to the kitchen. In one case, from the dining room, from my bedroom, from the basement, whatever. And when you say kitchen, you invoke all of those at once. You essentially invoke all of the potential behaviors that led to this more stable pattern here. And then when an input comes in, it's one particular one. That input might be, I'm in the bedroom. And it'll pick up one of those sequences exactly it'll basically say all these guys are depolarized. So it's a big union of the polarization. But now I do a very specific instance, here I am, and it'll essentially start playing back that motor sequence from that place. So I basically say, go to the kitchen is a high level, I'm using these as analogies, is a high level behavior I want to achieve. I now invoke a union of all the behaviors that ever could possibly do that. And then I have a specific input that comes in and says, you are here. It's like saying, you're at this note in this melody, and I know exactly, and immediately just start saying, I know exactly what to do to play it out. And it's sort of a union of these things, and you can walk through the depolarizations and so on. So this I'm pretty confident about. There's this, basically you're invoking a union of behaviors, sequences of behaviors, that then when a particular input comes in, you just know exactly what to do. And it just follows that behavior. As long as this thing is stable here, then it just follows that. So that's what's going on with these layer 5 cells projecting up into layer 1. And the similar things happening in layer 3 cells project up into layer 1. What's that going on there? That's sort of saying, I'm expecting you to see something, or I'm expecting you to perceive something, like look for the dog, or look for the cat. And you invoke essentially a union of layer 3 cells that are all depolarized. That could be all the different possible patterns that you might evoke with that thing. So now when an input comes in, even if it's ambiguous, it will be interpreted in that context. You'll immediately go to you'll go to that sparse representation immediately. You come in and say, I'm not really sure what this is, but because you've depolarized a set of these cells, you'll come right immediately to the sparse representation saying, yep, there's a cat, and you'll be biased to seeing the cat. So this basic idea of feedback from a stability to a changing pattern is happening both, and this is exactly what's happening. It's happening to layer 3 cells, and it's happening to layer 5 cells. Again, sort of suggesting this parallel construction between layer 3 and 5, where layer 3 is really a sensory pattern, and layer 5 is a motor pattern. And so you're invoking a sequence of motor behaviors, and this is essentially you're invoking a sequence of inferences, if you will. So this is one of those pieces that is clearly happening. But I still don't have the whole picture wrapped up yet. I'm still struggling how to put all the pieces together, and it's a multimodal problem with lots of different edges to it. So I think maybe, unless you want to keep talking about this.
I think we should let some other people have a chance to ask good questions.
Yeah, okay. Yeah. No.
Jeff, can you talk a little bit about your understanding of inhibitory cells and their role in temporal pooling?
In the role in temporal pooling specifically?
Yeah, you mentioned that on the mailing list.
Yeah, let me just think about that. So we're still working on exactly how we think temporal pooling is working. So that's the hardest part, maybe to jump into. How about I just give you a brief introduction to some inhibitory cell stuff, and then we can talk about some of which Virgil just mentioned here. So just as a general rule, 75% of the cells in the cortex are excitatory cells, and 25% are inhibitory. As another general rule, it is generally believed that the inhibitory cells are not where learning occurs. And there's a lot of reasons to suggest this. This is, you're shaking your head if you don't believe it, but This is general neuroscience belief. It could be wrong, right? We kind of subscribe to that. There's a lot of biological evidence to suggest this is true, in the sense that when you look at connections between excitatory cells, they're very, very sparse. Like you almost never see one. Very rare to see an excitatory cell make multiple synapses on another one, but it's really common in inhibitory cells. So in inhibitory cells, if I look at a column of excitatory cells, which I'll draw as brown here, and then you look at an inhibitory cell, which I'll draw as green, an inhibitory cell might connect many, many synapses to all these guys in ways that would just completely shut them down. There's no integration, there's nothing going on there. The inhibitory cells, they impact on a neuron in three places. They can form synapses on the cell body itself, which excitatory synapses never do. Only inhibitory synapses. Excitatory synapses are always on dendrites. They can also form synapses on what they call the very beginning of the axon. There's a little place in the beginning of the axon where the actual action potentials start, and they form synapses there, which is a very strong, basically saying, I don't care how depolarized you are, you're not going to generate a spike. It's not going to propagate. You're just not going to get one. So these are multiple ones from inhibitory cells. They don't seem to be carrying any information other than just shut down. And the third place they occur is along the dendrites, typically even the distal dendrites. And I believe those are actually changing the threshold of the distal dendrites, but we don't model that. So this is the way that the cortex can turn up prediction and turn down prediction. So I'm not doing enough prediction, or I'm doing too much prediction. Anyway, so inhibitory cells in general are believed not to be information carrying. There's a lot of reasons for that. There are many classes of inhibitory cells. It depends on who's counting, but you might think of like 5 of them or something like that. The most common one is the ones I've just shown you here, that when a cell becomes active, it activates an inhibitory cell and it tries to shut down everybody in some region nearby. Very strong, very fast. We rely on this for our sparse activation. This is essentially how sparse activation occurs. Very well documented. There are other inhibitory cells which have a very strong collimer aspect to them. And so they literally define these bundles of inhibitory axons, literally surrounding the mini columns. And there's 2 classes of those, and we're relying on them in a very fuzzy way to say basically, well, what they seem to be doing, at least some people have said, is that these cells inhibit these cells. So so this column one would inhibit the other ones, which essentially says, well, no one's inhibited anymore. All these cells here are not inhibited. This is what would lead to the collimer activation, is because you have fast inhibitory of inhibitory cells in a collimer fashion. And so those are sort of the general ideas, and there's lots of other flavors which are complex and things going on. So I just said here, we rely on these 2 basic ideas predominantly in both the spatial pooling function of selecting the column and then selecting one of the cells to win, just like Virgil was talking about. So, but we think what happens is even if the inhibitory column activation occurs quickly, one of these cells will start firing, and as soon as it starts firing, it then will inhibit the other guys. So there. Now, you ask specific in terms of temporal pooling. All right, I'm dancing around it because I'm not sure what I'm going to say about that. Let's talk about, we are still working through temporal pooling. We're still trying to figure out how we want it to work and how we think it works. And we've been modeling it mostly on layer 3 and 4. And so we're basically saying here, we're saying that if I have an unpredicted input here, that's the easy one. If I have an unpredicted input in layer 4, I get bursting columns. We believe that actually just propagates straight up into layer 3, and so you get bursting columns here. It's like nothing's happened. It's just like we're modeling layer 3. No temporal pooling going on. The challenge is when you have a good predictive model in layer 4, then you have a very sparse activation, and then another sparse activation, another sparse activation. Now you want to lead to a stable pattern up here, and as Sergey was just saying, you want to have as many of these changing patterns here map into that stable pattern. That's the basic idea. we started off, this is totally just, work in progress, so just bear with it. I might tell you something completely different a month from now. And I'm trying, maybe I'll be able to work back in your question about inhibition. Did you have a specific question about inhibition yet?
A little bit later, maybe.
Okay. Let me just start here. One question is, which cells am I going to be trying to, so when I say temporal pooling, we're talking about cells that have a proximal synapse here, dendrite here, and we want this cell to learn multiple patterns down here, pattern A, pattern B, pattern C, pattern D, pattern E, and things like that, right? And so right now we're testing the capacity of these guys, how many can a cell learn. We just did an experiment this last week or whatever, about 50. We think we can get much higher than that, but per cell, right? And then which cells are doing this? We originally said, okay, we're just going to have one cell in one of these sparse columns, one of the a sparse activation columns and one cell in each column. We're now working towards an idea that actually it might be more dense than that. We might have like one cell in all the different columns, or a more distributed, less sparse representation. There's some reasons for that. And so there would be a bunch of cells up here that are essentially staying on. So basically, they're winners, they're hysteresis, they just keep going until someone tells them to stop. And the signal to tell you to stop would be a bursting column. A bursting column says, you stop, because I got a new unpredicted input. But as long as I have an unpredicted input, I can keep trying these cells to keep learning. Now where does the inhibitory cells come into play there? At a very simple level, we're going to rely on these guys again here, because we essentially say, well, these guys will allow a very sparse representation, be active, and you'll have a bunch of cells that are winners, and they basically shut down anybody nearby, but you can still have quite a few cells, but it's just going to be a sparse representation. I'm not sure if we've thought about inhibitory cells anywhere else yet in that context. So maybe you have some specific thing you wanted to ask about.
Basically, layer 4 cells activate when they're unpredicted a number of layer 3 cells. I was wondering how that activation stops when the pattern is predicted. How only one cell is activated instead.
So the general rule we've been working so far is that when a column bursts, it's temporary, always.
Okay.
It's a transitory state. It's a temporary state where all these cells fire, but as soon as they start firing, one guy is going to win out. It's a winner take all because of the inhibition. So it's always a quick thing. I burst and then I settle on a sparse one. So that's always going to happen, and that would happen in layer 3 too. I would burst, and then I'm going to pick one. Right? It's just the cortex doesn't want to be in a bursty state. It doesn't like that. It's just a temporary thing. Does that help? No. Sure. Sure. Let's say you're sure with resignation. Like, oh no, not really. Well, I don't know. Look, we can walk through it if you want, but.
No.
Okay. If Subtar, if you think I'm missing the question somehow, just speak up and. Okay. I'll try another question or a follow-on to that one.
It's not really a follow-on, but it deals with layer 4.
Okay.
So, does sensory data come into layer 4 twice through both proximal and distal input, or is that just a implementation detail in the code?
Well, at the moment, you say it's an implementation detail. At the moment, when we're starting to model layer 4, we do not have sensory data coming into the distal dendrites.
The sensory motor, which is the union of the two.
It tries to predict, given the current sensory signal and the motor command you're about to make, what the sensory signal will be. So currently we do feed in the sensory and the motor into the distal.
Yeah, but not.
In layer 4, and then in the proximal.
Yeah. I'm sorry. Maybe I misinterpreted the question. So you have a cell here; it's getting feed forward, which we'll think about as sensory, right? And then on the distal guys, you're getting motor, and I guess you are getting sensory as well. Here's the interesting thing about it. What we didn't want to do initially is we didn't want to have other layer 4s connect here, because the other layer 4 cells represent a high order state. And that's what we need in layer 3. If you want to predict, from a high order state to a higher order state to a higher order state. But here, you don't want to do that because it's not a high order pattern. It's not a repeatable sequence. So we actually didn't want to have connections from other layer 4 cells, which is the only thing we have in layer 3. So here's what we do know. We know that something like 65% of the synapses on the distal synapses of a layer 4 cell come from, believe it or not, layer 6. What the hell's going on there? And we know that there is motor commands coming into the cortex through the thalamus, just like the sensory data, but we don't know where they go. I haven't been able to find anything about that. So we're predicting they're going to go there on the distal synapses. That's our prediction. Hopefully we'll be able to find some literature which proves that right or wrong, but that's what we're predicting there. And the idea behind this is, roughly, is we said, okay, we want to have, and I don't know where the state of our code is, but I wrote a little email about this at the beginning of this year. What you want to feed into these distal synapses is not necessarily the raw sensory data. You want to represent sort of the columnar representation here. It's you want to say, I'm looking at an eye, I'm about to move to the right, and I predict to see another eye. And I don't want to be at some high order state. I don't want to be saying, oh, I'm at an eye that I got to by looking up from the nose. I want to be able to say that's just an eye, I'm looking to the right. And so my original thinking is that you wouldn't want to take this raw sensory data. You'd really want to have a columnar a representation of it. And that could be coming from layer 6. You could go back and read that email. But a lot of this stuff is in flux here. So this sort of fit in the model. It says, okay, this is a non-high order state of the current input. This is the motor command. Then we can predict what's going to happen next. There are some complications here. We do know that even though 65% of the connections come from here, and we believe the motor connections are a lot of these as well, we do know that there's a few connections from other layer 4 cells. Not a lot, but there are some. We were starting to think about what those might be doing. So, that's the state of the knowledge. But this stuff, this number here and this here makes a lot of sense. And the fact that these cells are not highly interconnected makes a lot of sense. So that all fit with the theory. But we have a couple of things we don't understand exactly. We don't understand why there's some of these connections, and we haven't worked through all the details of how this is working. So we don't really model this. We just basically take the columnar representation and feed it into these distal dendrites. We're not modeling layer 6 per se. But it basically works in this context. So did I get close to answering your question?
Oh yeah, basically. I think it's an implementation detail. I think it's how you're getting first order sequence memory while still having multiple cells per column.
Yeah, well, we need the multiple cells per column to represent.
We actually do have multiple cells per column.
Right, and that was the.
We need that. We need that to create the high order sparse state that says, this is an eye I got to by moving to the right.
Yeah, it's not a hack. You still need that because you need to represent the spatial configuration that pattern is in.
Yeah.
It's just what Jeff said. It's not just an eye. It's an eye with a nose below it and a mouth even further below it. So that's different from other eyes.
Right. Sorry. Yeah.
The theory says you have to have multiple cells per column. The bursting, all that stuff is the same. The theory says, though, the distal dendrites need to be getting input from both a motor command and a non-high order spatial state. And the things we don't understand exactly is like, why are these other connections here? There's some small percentage are representing presumably a high order state. Why are they going there? We actually have a speculation perhaps why that's happening. And we don't really know what's going on in layer 6, but it's nice to know that at least it's getting input mostly from layer 6 and not from other layer 4 cells. If it was all from other layer 4 cells, we'd be in trouble. This wouldn't work. So we can speculate that layer 6 is sort of representing, and it is. These layer 6 cells are simple cells. So we can say, okay, it's getting some non-high order state from layer 6. And layer 6 gets direct, by the way, layer 6 gets direct feed forward sensory input. So so it's sort of like layer 6 is doing spatial pooling just like layer 4 is. And it's passing that representation up. So it's a bunch of stuff we don't really understand there. But overall, it hangs together really well. I mean, overall, this is very, very nice. Again, there's a lot of things that we speculate here that could turn out to be different, would have shot the whole thing to hell. But most of them fit pretty well. So there's a few things that don't fit very well.
We've got time for one more question.
Oh man, this is all went by so quickly. That's one more question for taping, but I'm happy to sit around and chat if anyone wants to do it. Another question? You've been waiting to ask the question.
So if you take voice, right? We got different temporal spaces that the brain is paying attention to, right? So you got like phonemes, you got like vowels, you
Yes.
got consonants,
Yes.
and then you got words, and then you got sentences,
Yeah.
and you got a
Yeah.
paragraph
Yeah.
or monologue.
Concepts.
And along the way, there are choices to be made as to how you pay attention to the time frame at which you want to create a hierarchy on. What might be the mechanism here as to which these different time spans?
If I understand your question, the question is, as you go up this hierarchy, and you use language as an example, but it could apply to anything,
Yeah.
right? You're going to be parsing this up in different sort of time epochs, like different links, and how does it decide, and what is that?
Yeah.
I think the answer is it's all organic. That every region or every layer in the cortex has capacity. It has some amount of capacity. It has capacity for sequences, and it has capacity for temporal pooling, and it does as much as it can. And then it says that's all I can do. And then it says, okay, the next guy picks over. If I could create a humongous, monstrous, super monstrous V1 or A1 auditory thing, it could learn a lot more than the ones we have. It could basically do more in that first region before you go up to the next region. But it turns out that there's a, how do I describe this? In theory, you could build a single layer neocortex that does everything. But it would take forever to train. You'd have to be exposed to everything. And we'd have an ungodly amount of memory required to do this. But if you have an infinite amount of time, or a very, very large amount of time, eons, and you had all this training data, you could basically build a monstrous, huge V1 or E1, and it can learn everything. But what happens is you can't do that. So what the cortex does is here's a general idea: is you have a region of cortex here. And it's getting, let's say, the primary auditory or primary visual, whatever. It's getting input, and it's getting this, of course, in a distributed fashion over some sensory organ. So it's getting a whole millions of fibers coming in, or tens of thousands, whatever. This guy, essentially, locally, it tries to learn as many sequences as it can. And the simpler they are, the longer they can get, the more it can learn. The more complex, the more of them are, the shorter they have to be, because it's a limited amount of memory. It's a fixed amount of memory. And it's literally topological like this. The cells can only learn within certain region, and they try to keep building longer and longer sequences, and then they say that there's no more memory here. So these guys learn as much as they can, and then you do temporal pooling over this, and then you represent these sequences as more stable patterns, and then they become the inputs to the next guy, and the next guy does the same thing, and it's trying to learn, and it learns longer sequences. And then you do this over and over again. So where would the words appear in here? Well, it depends on how much memory you have and how complex the world is. I don't think there's like, words are at this level and concepts are at this level and phonemes are at this level. It's not going to be like that. It's going to be wherever they kind of naturally max out. And the hierarchy basically makes this solvable in real time with limited amounts of memory. But you do lose a capability by using hierarchy. So the short answer to your question is, there won't be a sharp break for things like you and I think about, like words or phonemes. And I could give you lots of examples, like here's the phrase, good morning. Is that one word or two words? Right? It depends. If you hear it enough, it becomes one word. If you haven't heard it much, it's two words.
So if I give a long vowel, right? I mean, would that?
Think about it as the number of transitions, not the length of it. Okay, right? If I just sit here and go, that's not a sequence. Okay, don't think of it that way. Imagine you could break it up into little sound transitions. All right, so then don't worry about how long it takes to make those transitions. It's just that's the number of transitions that are comprised in this auditory stream, and then it'll just parse them up wherever it can.
I think the timing itself is done separately.
The timing is done separately. I've talked about that other times. The exact timing, how that would do. But that wasn't your question, I don't think. I think your question is like, where in the hierarchy, how would you decide where to parse up these things?
It would seem like there would be some sort of a state machine that it kind of keeps track of vowels.
No, I don't think so. I don't think so. You remember, cortex doesn't know anything. It's just looking at sequences. And this auditory cortex, or whatever cortex you're talking about, is no different than any other cortex. It doesn't know what this is. All it can do is look at the statistics of the data and say, all right, what are the repeatable sequences here? Where, how many of them can I learn before I run out of memory? How long can I learn before I run out of memory? And then, so I could imagine, for example, imagine I have a language. We came up with a language that has very few phonemes, and they're very simple, and the words are very, have a perfect architecture to them, Esperanto or something, I don't know what it is. And you would end up with a very different representation here than if I had some other thing where I had 50,000 phonemes, and it just makes it up as it goes. It just says, I'm just going to try to learn as much as I can, as much as I can, as much as I can, and that's it. I don't think there's nobody sitting there going, oh, this is language. Here's a phoneme, and here's a word, and I gave you an example of a word like good day or good day. Is it really one word or two words? Right, and you start out, it sounds like two words. It's good day, but then it becomes one word. And same with phonemes; they all kind of move together. You learn phrases, and there's no clear break here. We impose those things after the fact to try to describe what language is. That's what linguists do, right? They can say, oh, these are words and these are nouns. And this is why linguists have such a hard trouble understanding language, because they want to put the structure on it. In reality, that structure doesn't really exist.
And also, the more you learn, the more of these things, like good day, literally starts off as two sequences.
Yeah, so two words.
Then it becomes one sequence.
That's right, that's my point.
Because you never use them separately.
Yeah, yeah. Well, although good and day are still separate words.
Yes.
In my mind, I have a little bit this notion that if you're a baby and you don't know language yet and you're learning language, it's kind of like the frequency at which components occur independent of each other, like vowels are going to occur very frequently in many words.
Yes.
You're going to create a hierarchy around those, which will make it easier.
Well, first of all, vowels and consonants don't exist in speech. Phonemes exist in speech, right? Just to be clear. So,
right.
But yeah, that's true. Look, but we try to teach kids the structure to language, but that's not how they learn it, right? I think the first word the second word my daughter learned was strawberry. And because she loves strawberries, and she didn't think of that was just a thing. That was it wasn't to her mind. That's a 3-syllable word. No,
yeah.
She didn't. It was just one thing, strawberry, and then she probably didn't make a connection to any other words to have those. Over time, this all gets sorted out. But I don't think the visual world is not broken up that way. The tactile somatic sensory world is not broken up the way. The auditory world is not broken up that way. We impose language structure after the fact. We don't learn to speak by grammar rules. We learn to speak just by repeating what someone says and just hearing it again and again. And later we say, well, what was language composed of? It's composed of these elements and blah, blah, blah. It's not really true.
It's an observation of an apparent structure.
Yeah.
That just exists on its own, right?
Yeah.
The language just is what works for this.
The language is what works. It is what works, right? I mean, the same thing with music, right? You can break down music and you could say, oh, these are different types of phrases. If you take music theory, these are different sort of resolutions and chord progressions and different types of dissonance and so on. But in reality, the first musicians didn't do any of that. They just started playing stuff that they liked to hear, and then that was that. Then later we can go back and say, oh no, that is a such and such progression, and you resolve the seventh to the tonic, and blah blah blah. But they didn't start. No one started that way, and the language is like that too. We just started talking and making sounds and noises, and then later we come back and say, oh, let's put some structure on that.
Now, there are mockingbirds. There are these birds that will listen to a stream of audio.
Yeah.
And then they will echo, even throw in an ambulance on top of something else, and it's like a very precise, almost like a recorded it.
Yeah.
And it spits it out.
Yeah, they have no concept of structure in it. They're just recording the sequence.
So that means its neurons have to have really fast learning capabilities to stream.
Yeah. So we'll leave. I think Matt wants me to end. So I'll leave you with this thought. I was talking to the. What did you say? No, not end.
Had someone.
I'm sorry, we don't have time. We got demos in less than an hour.
Okay, I can keep talking. You can just stop recording.
Okay.
All right.
Say goodbye.
Bye. So if you want to keep talking about this, fine. I don't care. This thing is no longer green. Did it record?
No. Obviously, it died.
It's not green anymore.
That's okay. I have audio from the camera and the overhead speakers.
Okay.
Anyway. Oh yeah, it died.